Introduce Professor Sharon Chen, who's the Director of the Centre for Infectious Diseases in the Micro Laboratory Services at Westmead Hospital and for New South Wales. Sharon has a long-standing interest in fungal infections spanning diagnostics and epidemiology, and she's led many trials across these areas. And she is involved in national antifungal drug trials as well. She's a founding member of the Australian and New Zealand Mycosis Interest Group and very highly respected and accomplished mycologist. So over to you now, Sharon, to talk about drug resistance in the ICU.
Thank you, Monica. And goodness knows where you got that paragraph about me, but yeah, thank you for your very kind words. And thanks to all the organizers for having me on board today. So I've been asked to talk about drug resistance in the ICU, but obviously drug resistance doesn't apply only to the ICU. So I hope what I'm going to say is going to be useful for all those present. These are my disclosures, and I work at Westmead in Sydney. So what I thought today is to take it further. I mean, we've had a great introduction by Lewis and talk a little bit about drug resistance, not so much in N. glabratus, but in C. tropicalis. And I'll tell you why in a minute, and C. parapsilosis. And this stemmed from an outbreak of C. parapsilosis in a Sydney ICU that I was lucky enough to be involved in and to touch on the Aussie experience. And it's interesting to note that the WHO in 2022 classified all the Candida species that we've talked about today, glabratus, tropicalis, and parapsilosis, in the high priority group, with C. auris being in the critical priority group. So the first thing to note is that this goes back to 2014, and you can say, oh, these data are, a decade old, but I know that Abby Douglas and others are working very hard to update the scenario of candidemia in Australia in the 2020s. In the 2014 study, however, we note that whilst Candida albicans still reigned supreme, glabratus caused about 26.7% of infections, parapsilosis 16.5%, and tropicalis a small percentage of infections, 1/3 of whom affected patients in ICU. However, tropicalis was associated with higher mortality, 37 to 60%, compared with 31% overall for all candidemias. And what we were alarmed to observe was that the resistance rate to fluconazole rose from less than 2% 10 years ago, before the study, to 16.7% over that time period. And this was not due to a laboratory error, as you might expect, difficult reading plates, over-enthusiasm in calling resistance. This was proved by genetic studies. So just to take one step back, how would Candida tropicalis become resistant? Here's the fungal cell wall, here's the cell membrane, and here we have the nucleus, the mitochondria, and all the organelles. And the commonest mechanism of resistance is gene mutations in the ERG11 gene, which codes for ergosterol synthesis, a very important gene. But there are other important genes as well, such as those that encode for drug efflux transporters, such as CDR1 and MDR1. And we have genes that code for the regulators of the drug transporters, so upstream, and these genes are also important. And then we have the phenomenon of increased copy numbers and loss of heterozygosity in a diploid organism such as C. tropicalis, which can lead to azole resistance. We were in a position where we had, good collaboration with our Singapore colleagues and had a, whole cohort of fluconazole susceptible and resistant C. tropicalis isolates. And therefore, we sort of thought we should just check out the correlation between phenotypic resistance and genotypic resistance. Most of these isolates were bloodstream isolates, but there were some others as well. And in total, we had 17 fluconazole non-susceptible isolates. So I know the slide is busy, and the color codes: green are susceptible, red is resistant or non-susceptible, and the orange or brown is non-wild type. And you don't have to read all the numbers, but you can see with the red isolates, so over here, they had well-defined mutations in the ERG11 gene. So these two that you might expect from your review of the literature. There were 3 isolates, however, that did not have these mutations. There's one here, there's one here, and there's one here. So there's other mechanisms of resistance that one should worry about. When you look into greater detail or greater depth, you can find that apart from the ERG11 mutations, which are shown in pink, there are other mutations that co-occur, leading to significant amino acid substitutions in the TAC1 gene, which is here, and in the STB5 gene, which is here. There were some isolates who also had mutations in the CDR1 gene, but with CDR1 gene, it's not really about the mutations, it's about the overexpression, and we did not study this in detail. Now, there are heaps and heaps of genes. You can see that, I could list another 20, but in in diploid organisms like tropicalis, it's all about whether they are heterozygous or they occur in a homozygous state. And if you look at the fluconazole non-susceptible isolates, you can see in the red part of the Venn diagram that homozygous mutations, there were only 6 that were associated with phenotypic resistance. So, phenotypic susceptibility testing is still really, really important. So in summary, just to conclude this bit about tropicalis, it's a high-impact pathogen, including in ICU. Azole resistance is increasing, often mediated by SNPs in ERG11, but other mechanisms of resistance also have been documented. And the study of resistance by genomics is complicated by the fact there are multiple copies of each chromosome and by genome plasticity. Hence, assessment of SNPs requires careful thinking and assessment. Moving on to parapsilosis. So once again, I was lucky enough to be involved in this story with Minoshi Pereira, who was our registrar at that time. In a tertiary hospital in metropolitan Sydney, there's an ICU, 4 adjacent 15-ward beds. Each bed is in a single room and has access to a shared common central area. Between 2023 and 2024, with some surprise, 7 fluconazole-resistant C. parapsilosis isolates were observed from 6 patients who were either deemed to be colonized or infected. This is the story of patient 1, and I won't be going through all the stories of the patients this afternoon. 35-year-old lady who had conditioned to be in hospital, had a couple of wounds, had oozing wounds from the groin, the abdomen, and these were swabbed, as one would normally do, and C. parapsilosis was grown. No susceptibility testing was performed on the original isolates, but when this was repeated, in retrospect and after the ID consult, susceptibility testing was performed retrospectively. Now, these patients all had risk factors, as Lewis has described, CBDs, broad-spectrum antibiotics, prior antifungals, so and so on. Half of the patients died, and those that lived were cured with antifungal. Now, this slide also has very small writing, but just to illustrate a few points. So Candida parapsilosis infections or colonization occurred at a median of 34 days from hospital admission to a positive culture. for a median of 21 days from ICU admission to a positive culture. So quite long into the admission. And all these patients, all 6 of them, had an intravascular catheter device and had classic risk factors for candidemia. All the isolates from these 6 patients were fluconazole resistant as defined by the CLSI, and all underwent sequencing and had these mutations that were already well described in the literature, Y132F and D44Y in the TAC1 gene. There are other mutations, but as you can see from the C. tropicalis Venn diagram, some of these also occur in fluconazole susceptible isolates, or they occurred in the heterozygous position and therefore were not considered to be clinically significant. At the same time, and this is going back in time to 2017, we were lucky enough to have access to parapsilosis isolates from corneal transplant patients who contracted infection from donated tissue. There were 6 or 7 patients, and the isolates were also available for sequencing. So we had a problem. Parapsilosis is a diploid genome. It's very large. There is a reference genome, but it wasn't a perfect fit. Therefore, we used 2 pipelines, one which masks and one which doesn't mask, to have a look at the sequence, the whole genome sequences of these isolates, and did some simple recombination analyses and looked at various antifungal resistant databases that were available at that time. And I'm not going to show you all the results, but here's a simple SNP matrix using the in-house pipeline. You can see that this is the ICU cluster, this is the I cluster on top here, and everything else is in between. And naturally, the clustered isolates have fewer SNPs between them as compared to the so-called control isolates. But unlike a bacterial genome, where you might expect 0 to 2 SNPs between the related isolates, they were clearly separated by hundreds of SNPs. And speaking to a bacteriologist expert, they would say, oh, of course they're not related. It's too far apart. But they actually were. And here again, if you look at the tree, I sequences, ICU sequences, clearly more related to each other than to the other isolates. The problem for us was that the expected degree of genomic variation in outbreaks of C. parapsilosis is not well described. It's quite a clonal species. Therefore, the outbreak definition of each species of Candida may need to be personalized. And here in the interest. So to conclude, all Candida species are important, but they are not created equal. Genetic tools are maturing, not as mature as for bacteria or for viruses, and can complement our infection control and clinical staff and can assist in AMR and cluster scenarios. But I do believe that careful thinking and personalization of specific contexts is needed. With that, thank you very much. I do have one acknowledgement slide to thank my colleagues for the tropicalis work here, for the parapsilosis work here, and of course to Katrina Halliday, Alice Gordon, and last but not least, Vitaly Sinchenko, who have lent me all their expertise. Thank you very much.